हेलो क्लास, वेलकम टू बायोकैटलिस्ट। तो आज हम लोग सेल साइकिल एंड सेल डिवीजन का नेक्स्ट पार्ट कवर करेंगे एंड आज प्रोबेबली ये चैप्टर कंप्लीट हो जाएगा। तो चलिए शुरू करते हैं। Meiosis. The production of offspring by sexual reproduction includes the fission of two gametes, each with a complete haploid set of chromosome. Gametes are formed from specialized diploid cell. The specialized kind of the division that reduces the chromosome number by half results in the production of the haploid daughter cell. This kind of division is called meiosis. Meiosis ensures the production of the haploid phase in the cell life cycle of sexually reproducing organism, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of the haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of the nuclear and cell division, which is called as meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Meiosis 1 is initiated after parental chromosomes have replicated to produce identical sister chromatid at the S phase. Meiosis involves pairing of the homozygous chromosomes and ribosomes between the non-sister chromatids of the homologous chromosome. Four haploid cells are formed at the end of the meiosis 2. Kitna? 4, okay? Meiosis events can be grouped under the following: meiosis 1 and meiosis 2, which have prophase 1 and prophase 2, metaphase 1, anaphase 1, telophase 1, and have 2 in respect of it. Meiosis 1, which have prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into following 5 phases based on the chromosomal behavior, that is liptotin, zygotin, pachytin, diplotin, and dikinase. During liptotin stage, the chromosome became gradually visible under light microscope. The compaction of the chromosome continues throughout liptotin. This is followed by the second stage of prophase 1, that is zygotin. During this phase, chromosomes starts pairing together, and this process of association is called a synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate the chromosome synapsis, which is accompanied by the formation of the complex structure called as synaptial complex. This complex is formed by a pair of the synapse to homologous chromosome, which is bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of the prophase 1 are relatively short-lived compared to the next stage, that is pachytin. During this stage, the four chromatids of each bivalent chromosome become distinct and clearly appear as a tetrad. This is the stage which is characterized by the appearance of recombination of the nodules. The sites at which the crossing over occurs between the non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of the genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called as recombinase. Crossing over leads to recombination of the genetic material on the two chromosomes. Recombination between the homologous chromosome is completed by the end of pachytin, leaving the chromosome linked at the sites of the crossing over. The beginning of diplotin is recognized by the dissolution of the synaptial complex and the tendency of the recombinant homologous chromosome by the bivalent in order to separate from each other, except at the site of crossover. These X-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiotic prophase 1 is dikinase. This is marked by the terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of dikinase, the nucleolus disappears and the nuclear envelope also breaks down. Dikinase represents a transition to metaphase. Metaphase 1. The bivalent chromosome align on the equatorial plate. The microtubules from the opposite poles of the spindle attach to the kinetochore of the homologous chromosome. Here is the picture of it. You will go through the detailed study of the different phases which is mentioned over here. Just go through it, okay? Now let's move to anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centromes. Here we can see the exact thing from the following picture. No, telophase 1. The nuclear membrane and nucleolus reappear. Cytokinesis follows, and this is called as the dyad of the cell. Although in many cases the chromosome to undergo some dispersion, they do not reach extremely extended state of the interphase nucleus. The stage between the two mitotic division is called interkinase and it is generally short-lived. There is no replication of the DNA during interkinesis. Interkinesis is followed by prophase 2 as much simpler than the prophase 1. So let's go to prophase 2 which is a subpart of the meiosis 2. Okay. So prophase 2. Meiosis 2 is initially immediately after cytokinase, usually before the chromosome have a fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of the prophase 2. The chromosomes again became impact. Metaphase 2, at this stage, chromosome align at the equatorial tubules, that is the kinetochore of the sister chromatid. Anaphase 2, it begins with the simultaneous splitting of the centromere at each chromosome, which is the holding the sister chromatids all together, allowing them to move toward the opposite poles of the cell by shortening of the microtubule attached to the kinetochores. And here is the figure 2 of the meiosis 2. Just go through this figure also. Telophase 2. Meiosis ends with telophase 2, in which two groups of the chromosome once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of the tetrad of cell, that is, 4 haploid daughter cells. Now, the significance of meiosis, which is the last topic of this chapter. Meiosis is the mechanism by which a conservation of the specific chromosome number of each species is achieved across a generation in sexually reproducing organism, even though the process per se paradoxically results in reduction of the chromosome number by half. It also increases the genetic variability in the population of the organism from one generation to the next. Variations are very important for the process of evolution, and evolution के बारे में हम लोग डिटेल्स में पढ़ेंगे 12th में. सो इस तरीके से हमारा सेल साइकिल एंड सेल डिवीजन भी कंप्लीट हो चुका है. एंड मैं बता दूं कि इस चैप्टर का जो 2025 का क्या यार होता ही नहीं है कुछ. तो मैं बोल रही थी कि इस चैप्टर में 2025 का जो है ना, क्या हो रहा है यार, गुस्सा आता है ना. सो 2025 का इस चैप्टर का जो PYQ है वो मैंने कंप्लीट कर दिया. 2025 में सिर्फ एक ही PYQ आया है, वो शॉर्ट वीडियो में है. एंड बाकी ईयर्स के PYQ मैं एक लॉन्ग वीडियो लाऊंगी, उसमें कंप्लीट हो जाएगा ये PYQ भी आएगा. एंड ऑल्सो एक लाइक इस चैप्टर के एक्सपेक्टेड क्वेश्चंस का भी सीरीज़ आने वाला है, और टॉप MCQs भी इस चैप्टर के बेसिस में आने वाला है, तो देख लीजिए. एंड सेल चैप्टर का भी सारा कुछ कंप्लीट हो चुका है, तो प्लीज़ वो भी देख लीजिएगा. 2025 का पूरा PYQ का सीरीज़ चल रहा है हमारे शॉर्ट वीडियो में, तो देखते रहिएगा. तो आई विल कम विद द नेक्स्ट चैप्टर विद अ फ्रेश मोड. सो तब तक के लिए अच्छे से पढ़ते रहिए. बाय बाय.